Concept 2, we are going to talk about protein synthesis, and I'm so excited because your proteins are so important. And so this is a critical process because this is how your body and your cells in your body they make proteins. So, this is the process of reading the instructions that we have said are stored in your DNA, and we're going to make a polypeptide. Now, hopefully, you remember from Unit 1, Concept 3, when we learned about macromolecules, that the monomer of proteins is amino acids, and the polymer is a polypeptide. A polypeptide is a chain of amino acids linked together by peptide bonds that will then be folded into a protein. Remember, proteins have very complicated 4 levels of structure. Their first primary structure is the sequence of amino acids that makes the polypeptide chain. Then that gets folded into a beta sheet or twisted into an alpha helix, which then is either bent or folded into a more three-dimensional shape, which then combines with 2 or more polypeptide chains to form the final protein structure. Remember, form dictates function, meaning the shape determines what the protein does, and proteins are the most diverse macromolecule. So it makes sense that they would have such diverse and variety, so much variation in their structure so that they can do all the things that they do. So today we're going to learn just about level 1, primary structure. How do your cells read your DNA and figure out the order of the amino acids in the polypeptide chain? So I may refer to making a protein or making a polypeptide. Literally, we're making a polypeptide. But remember, that polypeptide will hopefully go on to be folded in to make a protein. So if I refer to either or, now you know what I'm talking about. Now, the central dogma of genetics, this is like the big overarching idea that is so critical to understanding this unit. Now, before I get to it, we need to refresh some memory. Here's a picture of your cell, and here it's just showing the nucleus right here. Remember, your DNA is the instructions for making you who you are and making the proteins that run your show. It is located in your nucleus, which it cannot leave because we learned in Concept 1 it has this very bulky double helix structure. But if you remember from Unit 2 cells, when we learned about organelles, proteins are made in ribosomes. Ribosomes are stuck on the rough ER or floating in the cytoplasm. So we have an issue. The instructions are in one location, and then the actual result is in another location. So, this process of protein synthesis takes 2 steps. First, we have to make a copy of the DNA that is small enough to leave the nucleus and go to the ribosome, and that is called RNA. If you remember, RNA is this skinny little structure. It has a single strand. It's much smaller. It is able to leave the nucleus and go to the ribosomes. Then, we will translate it into a protein. We will take the instructions that are in the language of nucleic acids and translate them into the instructions of protein. All right, so this is the central dogma down here, DNA to RNA to protein. So we're going to walk through these 2 processes of transcription and translation in these notes. Now, before we can do that, I need to highlight there are lots of types of RNA, but there are 3 essential types you need to know. First is the messenger RNA, and it is specifically written lowercase m and then uppercase RNA, mRNA. This is what you visualize and what I showed you in Concept 1 when we saw the single strand of RNA. That's mRNA. It copies the instructions from DNA, and it's small enough then to take that copy of the instructions out of the nucleus and into the cytoplasm where the ribosomes are. Now, tRNA is very important too. Think of it as transfer. It stands for transfer RNA, but I also think of it like a taxi. It is going to read, it's based on whatever this mRNA says, whatever the message says, it will bring the amino acid. So this is like the instruction manual, and this is the ingredients that are being brought and dropped off. So it transfers amino acids to the ribosome where we're assembling the polypeptide chain that will make the protein. And then you also have rRNA or ribosomal RNA. This, along with proteins, makes up your ribosomes. It's made in the nucleolus. You may remember that from Unit 2 cells. And it also helps to catalyze the formation of peptide bonds between the amino acids that will make the polypeptide chain. So first, transcription. If you've ever gone to the counselor at school and asked for a copy of your grades, you've gotten a transcript, which is just a copy. So we're going to make a copy of the important part of the DNA in the form of mRNA. So the overall process is DNA to RNA, but specifically mRNA or messenger RNA. So the purpose is to carry the code or carry the instructions out of the nucleus because DNA is stuck there and proteins are made in the ribosomes. Where does this go down? It happens in the nucleus because that's where the DNA is. We're going to start with DNA and we're going to end with a small strand of mRNA. So what happens? First, the enzyme RNA polymerase binds to something called the DNA promoter, and that is where transcription will begin. That's where it knows where to unzip the gene that needs to be copied. So this is very different from DNA replication, which is going to copy the entire strand of DNA. Here in transcription, we're literally only going to unzip and copy the gene that we need. Remember, there can be thousands of genes on one chromosome. So we're just going to copy the one section that we need. It's very efficient. And what it's looking for, the RNA polymerase is looking for the TATA box as a signal. It's just a bunch of TAs that signals, okay, this is where you would start transcribing. Once it locates that and binds and unzips, the RNA polymerase is then going to use complementary base pairing rules to match RNA nucleotides to DNA. So remember, C still goes with G, but in RNA, A goes with U, because there are no thymines in RNA. Then it'll release the completed mRNA when it's done copying that specific gene, and the DNA will zip back up. And then the mRNA leaves the nucleus and enters the cytoplasm where it can be translated. So again, DNA is copied into a complementary strand of mRNA in this process. Now, the DNA template is read in the 3 to 5 direction, so we're going to make RNA in the 5 to 3 direction. So we're going to take that DNA, we're going to unzip it, and then we're going to make our mRNA, and then the DNA zips back up. So we have our DNA and then our separate mRNA strand. So here's an example of that. Let's say this is the gene sequence on the DNA. It's the template. The 3 to 5 sides are template. The mRNA is going to be whatever corresponds to that. So T will go with A. A, because we're making RNA, goes with U. C goes with G, G goes with C, etc. And so that's how we figure out the mRNA sequence. This is my copy of the DNA's instructions. Now, after transcription, there are some modifications that happen to this mRNA before it moves on. Technically, at the end of transcription, you've only made pre-mRNA, but for our purposes, you can just think of it as mRNA. So one of the main modifications that's happening is called RNA splicing. mRNA has introns, which are non-coding regions, and it has exons, which are coding regions. I think of introns as interrupting the sequence, and exons are the expressed regions. They're what we actually need instruction-wise. And so what RNA splicing is going to do is it's just going to cut out. It's going to snip out those introns and splice together those exons and get just the pieces that we need together. Now, based on how this goes down, you can make a ton of different combinations of exons and many polypeptides just from one gene, depending on how you splice them together. And so this is one of the ways that we can make there's so much variation in what can be created just from one set of instructions from the DNA. Now, another thing that happens is the 5' cap. This is also referred to as a G cap. It's a modified guanine, remember that nucleotide, and basically it just gets stuck on the end of the 5' end of the RNA. And this 5' cap, this G cap, it helps the mRNA bind to the ribosome when it's going to get there for the next step. Also, a poly A tail is added. Poly means many. So 50 to around 520 adenines are going to be added to the 3' end. Now, why would we do this? This just helps the mRNA get out of the nucleus. So these are 3 examples of different modifications that are happening to that mRNA after it's copied before it leaves the nucleus and goes on to be translated. Now, before we can get in translation, there are some key words we need to know. One is the genetic code. This is the code of instructions for how to make proteins. So you are about to be a decoder. You are about to be a translator. You are going to translate RNA or nucleic acid language in the form of mRNA to protein language in the form of amino acids. And you're going to do that by using the genetic code. You will be given this on all assessments to reference. Now, sometimes the format of how this is displayed is different, but I really like the wheel. We're going to read your mRNA, the messenger RNA, in threes, in triplets. So 3 nucleotides at a time. And 3 nucleotides is the same thing as a codon, a set of 3 nucleotides on the mRNA. And it's going to tell us which amino acid the tRNA should bring. So the tRNA has an anticodon on one end, which is just a complement to the codon, and it has the amino acid, the building block for making proteins, on the other end. And the amino acid corresponds to whatever this codon is. So, for example, let's say the codon says AAA. You start in the center and you pick the first letter. So let's pick A. And then you go to the second part of the circle, which is A. And then the third part, which is A. And that tells you lysine. So on the outside here, we have all 20 amino acids. And this is how we determine which amino acid is made into the polypeptide chain. So you always read from the inside out. So let's look up CAC. So starting the inside, C, and then A, and then C. That would code for histidine. Okay, so this will be given to you. This is the genetic code, and we'll refer to that. Okay, translation. We're going to take the mRNA and translate it into a polypeptide. So we're going to follow the instructions that were copied into the mRNA, and we're going to make a polypeptide, which will fold into a protein. This goes down in the ribosomes because that's where proteins are made. We will start with mRNA, and we will end with a polypeptide. Now, here's the process. mRNA attaches to the small subunit of the ribosome. The ribosome then starts reading, always in the 5 to 3 direction, and it always starts with the codon AUG. So if you can look down here on my mRNA, it always begins with AUG. I think that school starts in August down here where we live, so I think of August as the beginning of the year. It's the beginning of translation. Remember, 1 codon equals 3 RNA nucleotides. So then we're going to keep reading in threes. The tRNAs right here are going to act like taxis, and they're going to pick up amino acids and drop them off based on what the codons say. They're going to keep doing this, and the amino acids are going to be bound together with peptide bonds until we literally get to a codon that says stop. It will say stop on the wheel. Okay, I'll show you. Look up UGA. UGA codes for stop. There's 3 stop codons. So that's how you know when the process ends. And that's where we know that the polypeptide chain is complete. Now, these tRNAs, these anticodons on them have an important role. Because they're the complement to the codon, it's kind of our way, it's almost like a puzzle piece. Do they fit together? If they do, that means that the tRNA brought the right amino acid. If they don't, that means it brought the wrong one and it won't drop it off. So that's one way that this process has a built-in proofreading to it to prevent mistakes. So again, this is the process of interpreting the RNA's message to make a polypeptide into a polypeptide that'll then make up a protein. So we're in the ribosome here in the cell. This is zooming in. Here's that small subunit we said the mRNA attaches to. Here's a large subunit, and there's these different sites to it that I'm not going to get into. But basically, the tRNAs are dropping off the amino acids, and they're getting bonded together to form this polypeptide. And I have a great animation I'll show you when we're gathered together so that you can kind of visualize this better. And there's our genetic code. You'll always be able to reference it. So let's do an example together. Remember that DNA sequence that we had at the beginning? We already transcribed it into mRNA. Now we're going to translate it. So first thing you have to do is you have to find the AUG because that's where you'll begin. And from there you start dividing into threes because that's your codons. That AUG sets your reading frame where you will begin reading and translating. Now, I often just find it and then I just draw lines every 3 from there. But some people like to separate them out, so you can also do that, where every 3 you just start separating them out into the codons. That's fine too. Now, in this example, the AUG was right at the beginning, but it may not always be like that. So you got to make sure you scan to find it before you begin. Now we're going to translate. We look up the codon on the genetic code to figure out what the amino acid is. So first we have AUG. Go back to our code, start in the center, AUG. That codes for methionine. And so then when we go back to our example, that first amino acid is met for methionine. All right, even though I have the full answer here, let's look up CGA. Start at the center, CGA. That codes for arginine, ARG. And you'll keep going until it literally codes for stop. So let's go back to look up UAA. UAA codes for stop. That's how we know it ends. You will always know if you did it right because it should always begin with a met, because we always start at AUG, and it should always end with a stop. Now, at times, you may be asked to also look up the anticodon on the tRNA, because remember, those transfer RNAs have the amino acid on one end, and then they have an anticodon that's complementary to the codon on the other end. So what would be a complement to AUG? UAC. CGA would be GCU, which we see down here. And so you can see all these. Remember, it's tRNA, so there's no thymines down here, only uracils. Okay, let's review these processes that we've discussed over the last 2 concepts. DNA replication. The purpose is to make identical DNA strands during the S phase of interphase before the cell divides so that the resulting daughter cells have all of the genetic information in them. The DNA strands are made with nucleotides A, T, C, and G, and this is happening in the nucleus because that's where your DNA is located. The purpose of transcription is to make a strand of RNA from DNA that's small enough to leave the nucleus to be translated in the ribosome. We do this with the building blocks of the RNA nucleotides, which are AUCG, and this again happens in the nucleus because that's where DNA is. Translation, we're going to make a protein, a polypeptide, from the mRNA. And we're going to do that with tRNAs, which are carrying amino acids, the monomer of proteins. And this happens in ribosomes because that's where proteins are made. Now, if all of our body cells have all of our DNA in them, how come my cells are so different? So for example, why is a skin cell so different in function from a muscle cell or a brain cell if they all have the same DNA and thus they could all have the instructions for making all of the same proteins? Well, the answer is regulation. Gene expression, so which genes we're actually going to transcribe and translate or not, is so highly regulated. Some genes get turned off and some get turned on, and this is critical for cell differentiation. Your cells don't make every single protein. They only make the ones that they specifically need for their specific jobs. And this regulation is happening before, during, and after transcription and translation. There are so many examples of this, but one just simple example are transcription factors. These are regulatory proteins that control gene activity. Repressors either decrease transcription so it won't be happening, and activators increase it so it keeps happening. Another random example is HIV. That's a virus that actually can disrupt regulation and mess up the gene expression process. So there's so much that goes into this, but these are just a couple examples. I'll go in way more detail with my AP Bio students if you take that class with me. Okay, last but not least is epigenetics. This is a fascinating topic. This is the study of changes in gene expression that are heritable, meaning you can inherit them. But here's what's so important. This is different from a mutation. If you remember, a mutation is a change in the actual DNA sequence. So remember, we're talking a change in like the A's and the T's and the C's and the G's from the original DNA sequence. This is different. Epigenetics is not affecting the actual DNA. It's not affecting the actual instructions. It's affecting how those instructions get expressed, how that DNA gets expressed. So if we're going to actually transcribe and translate those instructions. So one example of epigenetics, and we'll learn about more in a web quest we're going to do together, but is histone modification. So remember, your DNA is super long and it can't fit into your cells unless we bunch it up really tightly and condense it. And so histones are proteins that the DNA coils around and it makes these nucleosomes, and that's what bunches up your DNA into chromosomes. Now, if the histones are really, really tightly winding the DNA, then we can't transcribe the DNA. But if it's relaxed, then the DNA is able to be transcribed. And so that's one example of gene expression, and epigenetics can affect that, whether or not the histones coil tightly or not, and allow the gene to basically be turned on or off to be transcribed or not. And that is your overview of protein synthesis.